climate change. You know, soil is one of the most important creation of the nature, and we cannot create or cannot make soils in laboratory. About 1,000 years are required to form a millimeter of soil, and it acts as a major reservoir of terrestrial carbon. About 760 petagram carbon is stored in the atmospheric pool, and it is about 4.5 times the soil carbon, more about 4.5 times than the biotic pool, that is what the trees or biosphere contains. It's about 2,550 petagram. Out of that, 1,550 petagram carbon is present in the form of organic carbon, and about 950 petagram about inorganic carbon. And this inorganic carbon is mainly present in calcium carbonate forms. So from this data, you can see the importance of soil, about a sink of carbon. So why we are interested in soil carbon? Mainly, it has been found that throughout the research using different long-term experiments as well as different warming experiments in Europe and US, soil organic carbon ensures global food security, and it acts as a nutrient reservoir of required by the plants. So in this way, it helps in mitigating the harmful effects of global warming and climate change. It mainly gives the production system gets resilience in different extreme climate events. And it has been found that about 20% of the targeted CO2 emission could be met by agriculture through soil carbon sequestration. Now, soil carbon sequestration, we often say, but it is not like that. Your whatever soil carbon concentration in the soil, whenever soil carbon concentration is increased, we say this is soil carbon sequestration. It's not like that. You know, in photosynthesis, the photosynthates are produced, and these photosynthates go after the plant requirement. All this photosynthesis goes to soil through roots in the form of rhizodeposition or root secretions, these things. So after that, it goes to different pathways through, such as physical, chemical, as well as biochemical stabilization. Physical means the carbonic material which are stored within the aggregates, both macro as well as micro aggregates. We say this is the physical protection. That means those organic or carbonic materials cannot be accessed by the soil enzymes. So we say this is the physically protected organic carbon. And secondly, there are sand, silt, and clay particles in the soils. Out of that, silt and clay particles are played an important role in binding those carbonic materials. We say this is the chemical stabilization, particularly the 1:1, not 1:1, mainly 2:1 or 2:1:1 layer silicate minerals within the silica tetrahedral and alumina octahedral layers. Carbonic material goes inside the layers. We say this is the intercalation of the organic matter. And so this is very important mechanism of carbon sequestration in soil, because this carbon is very stable. It cannot be oxidized easily by the microorganisms. And third one is the biochemical stabilization. That means the inherent chemical composition of the organic substance, what you are added, maybe it contains higher amount of lignin, polyphenol, that's etc. And it confers stabilization to the inherent organic material what we are adding. So soil carbon sequestration, these are the three mechanisms, and we can quantify these things by using different fractionation methods. And so whenever we say about soil carbon sequestration, these three mechanisms are very important. And soil carbon, particularly organic carbon, is very important. You can see through these slides. And if somehow we can increase the carbon concentration of the soil, almost 70 to 80% problem of the soils are solved. And particularly starting from soil physical properties to chemical as well as biological properties are mostly influenced by soil organic carbon. And there are so many ecosystem functions are performed by soil organic matter, starting from plant health improvement to soil aggregation, aeration, water retention. And these ecosystem functions are linked to support different important processes such as net primary productivity and ultimately the climate regulations. Now coming to agroforestry, we know agroforestry, there are agriculture, the two terms are associated here, one is agriculture and another one is forestry. And these are various management practices and it has potential positive implications for climate change mitigation because by both the means, agricultural crops and forestry crops will go simultaneously and both have significant influence on soil quality enhancement and particularly through increasing soil organic carbon. Just imagine it has been found that about 40 to 50% of the photosynthesis of cereal crops starting from rice, wheat and maize goes to soil through roots in the form of rhizodeposition or root secretions. Then imagine a tree species, maybe about a eucalyptus tree or prosopis tree or any mango, guava or any fruit trees about 10 years old. So how much photosynthesis of produced in the leaves can go to soil through the roots? It's huge. And till now, I have no data about that. But we are mainly concerned about the above ground biomass part. But yes, sir.
Your slide is not moving.
Not moving. Now? Now moving?
No, sir.
Now?
Yeah. Yes. Yeah.
Okay.
Yes, sir.
So we are mainly, first slide I have shown about the biosphere, that means the tree species which contained about 560 petagram carbon. And soil carbon contains 4.5 times of that. So the soil carbon is much more important than the above ground biomass. And from the above ground biomass, we mostly measure the carbon content in the form of, mainly through allometric equations. So we need DBH data as well as plant height data. From that, we calculate the above ground biomass carbon. But mostly the soil organic carbon in those trees or forest tree species, it is mostly ignored. And there is an importance of sampling up to at least minimum 2 meters soil depth because the roots of the trees goes much deeper than the cereal crops. So you can see about 3 to 4 times more biomass mostly are mostly found in agroforestry system compared to the traditional tree species or treeless cropping systems. And in Africa, this is an example, it constitutes about the third largest carbon sink after primary forest and long-term fallows. And in addition to that, the suitable area for agroforestry worldwide is much larger than the existing systems. And it has been found that a total of 1550 million hectares in Africa, in African continent, are suitable for some time of agroforestry. And these are some of the management practices which affect the carbon stock and the forest. Particularly recently, even last year, there are so many occurrences of fires in California, particularly the west coast of the United States. So fire management is very important. In India, forest fire is not very common. But in the US, it is very common. And then afforestation, then conversion of primary forest, and so on. These are some of the management practices. Stabilization or carbon accumulation, when the input is more than the output is mainly in the form of oxidation of organic carbon. And you can see there, these are some of the factors when any living or dead organisms, particularly litter, when you add any litter or forestry litters for agroforestry systems, the microorganisms start working on that. And after that, through the decomposition process, there are formation of labile organic carbons, and then it acts with different particles of soils, starting from sand, silt, clay to iron, aluminum, those things. And then there is a different environmental factors. And after that, the fate is mostly oxidation of organic carbon to CO2. And it has been found that about 10% of the organic carbon, particularly labile forms, are susceptible to leaching, and it goes to downward to the particularly at about around 2 meters soil depth. But it also depends on the clay content of the soil. If the soil is containing heavy clay, then the leaching will be less, and if it's sandy soil, then leaching will be more. And we want more and more carbon should go deeper in the layer, because that carbon is more stable. And agroforestry or forestry systems are efficient in converting or transferring more and more carbon to lower layers, because lower layer, the microorganisms activity are much lower, and also due to that the oxidation is less, and that carbon is more stable compared to the surface layer. This is some data what we have studied at CSS Karnal under this is under mango plantation. And these are the four pools we studied, and you can see at surface layer the very labile carbon concentration is high, more than 9 megagram per hectare compared to the lower depths. But you can see below 1 meter depth, still the very labile carbon concentration is near about 6.5 megagram per hectare, and this indicates the roots of the mango trees. It's about 30 years old mango tree, big mango trees. And these trees were planted long back in our sodic soils. That time the soils are sodic. Through auger hole technology, you might know auger hole technology for planting of trees, saplings about larger depths, and these are planted through auger hole about 1 meter soil depth. And because of the roots of the mango trees goes much deeper, below 1 meter soil depth, you can see the very labile carbon, that is which can easily be consumed by the microbes. And compared to the if you see the percentage of TOC and in active and passive pools, below up to 1 meter soil depth, the active pool was higher, and after that you can see the again around 1.2 meters soil depth, the active pool was higher. And these are some of the pools of carbon, because passive pool carbon is not oxidized easily by the microorganisms, and active pool carbon is readily consumed by the microorganisms, and it supplies also nutrient to the plant roots. And this is under guava plantation. It contains compared to the mango about 3 ton per hectare higher very labile carbon compared to the mango plantation. And but you can see below 1 meter depth, the non-labile carbon fraction is higher. And in the photo, you can see these are the calcium carbonate concretions at 1 meter soil depth under guava plantations. Because these soils are sodic long back, now these are reclaimed, and pH goes down to about 7.5, sometimes below 7. And we found about 6.8 pH under lychee plantations. This is under lychee, and it contains about 16 ton per hectare very labile carbon, and it is even higher than guava. And if you see the active pool and passive pool carbon concentration, there is a little bit higher passive pool, about 5.47 ton per hectare at below 1 meter soil depth compared to active pool. And if you see the percentage of TOC as well as the active pool and passive pool distribution, the active pool concentration throughout the depth was higher in lychee plantations. And this is interesting observation we can say, because the biomass leaf litter, particularly leaf litter is measured, and leaf litter is higher in lychee plantations compared to the other land uses. Why? Because in lychee there are the leaf fall what we measured, the leaf litter was significantly higher compared to the other plantations, and it might contribute to higher amount of very labile pool carbon that it is showing in terms of active pool carbon. Similarly, under Jamun also CGM Cumini, we studied different carbon pools, and it contains significantly higher, about 15 ton per hectare carbon in very labile fraction compared to the other pools. And below 1 meter depth, non-labile carbon concentration increased significantly, and it constitutes about 9 ton per hectare below 1.5 meter depth. And similarly, the distribution of active and passive pool showed quite interesting data. Below 1 meter depth, passive pool carbon concentration was higher in jamun plantations. Now coming to eucalyptus. Eucalyptus showed significantly higher very labile carbon, about more than 18 ton per hectare carbon at surface soil. But below 1 meter depth, again, non-labile carbon contain higher amount of eucalyptus plantation. And eucalyptus, significant observation where there are bark fall as well as the twigs are accumulated on the below the trees, and so this might have contributed higher very labile carbon at surface layer, but at lower depth the decomposition and also the composition, we have not studied the composition. Maybe higher amount of lignin or polyphenol content are found in maybe found in the leaf litter of the eucalyptus. And this is under Prosopis diliflora. About very labile and labile carbon concentration were similar at surface layer, but below 1.5 meters soil depth, the non-labile carbon concentration was higher compared to the other pools. And if we compared the depth-wise variation of passive pool carbon, and we found guava significantly contained higher amount of percent of total TOC of passive pools, and you can see about 75% of the TOC is present in passive pool under guava, and it resided between 0.5 and 1 meter soil depth compared to the other plantations. And if we compare different percent of TOC, you can see with depth, active pool carbon concentration decreased and passive pool carbon concentration increased, and significantly higher was observed at under eucalyptus. It's about 1 to 1.5 meters soil depth. Higher passive pool carbon concentration was observed under eucalyptus. And if we compare irrespective of depth, guava contained higher amount of about 133 ton per hectare carbon in total carbon. And if we see the active pool, the again higher amount of carbon was active pool carbon was observed in eucalyptus, but the passive pool carbon in guava was about 56 ton per hectare, and it is highest compared to the other land uses. We also studied the correlations among the carbon pools, and it was highest, means all the carbon pools are correlated to each other. And liability index we have calculated from using the different pools. If you see the under guava plantation, the liability index was lower compared to the other, up to 80 centimeters soil depth. But under jamun plantation, below 1 meter soil depth, significantly lower liability index was observed. Lower liability index higher means that carbon can be easily oxidized, and liability index lower means that cannot be easily oxidized, that is more stable. So under jamun, below 1 meter soil depth, stable carbon was found, and under guava, below 80 above 80 centimeters soil depth, the stable carbon concentration was higher under guava plantation. We also calculated the stratification ratio. Stratification ratio means the carbon concentration at surface layer divided by subsurface layer. And more higher stratification ratio means normally more than 2 indicates good soil quality. And we found under jamun, the surface soil contains good quality compared to the other plantations. And lower was observed under mango and guava plantation. This is one of the work of C.S. Sarai long back in 1990. Singh and Gill, they studied different organic carbon concentration under different tree species. And they found Prosopis diliflora contain higher amount of organic carbon. This is in gram per kg. And this is Waklan black carbon. I suggest whenever you study carbon concentration, also report the carbon stock, and for that you need bulk density data. So whenever you study or measure organic carbon in water black or any method, try to convert that in organic carbon stock by multiplying bulk density and soil depth. So this is very important. Only reporting in concentration in gram per kg is not sufficient. This is a recent study conducted by researchers of Punjab Agriculture University. They studied different agroforestry systems, carbon dioxide assimilation capacity, and they found the poplar and wheat-based system content showed higher amount of CO2 assimilation compared to single poplar as well as whole wheat system. And different up to 7 years old, they studied that. And this is one interesting observed study made by US scientists. We know tropical forest or tropical rainforest stored highest about 1/3 of the global SOC. Tropical rainforest, not temperate rainforest. And they studied different protected organic carbon, particularly mineral associated as well as particulate organic carbon. And land cover means what type of forest, whether it may be shrubland or boreal forest, temperate forest, or tropical forest or grassland, it significantly influences the mineral associated carbon. Mineral associated carbon means what I showed the mechanism of carbon sequestration earlier. The carbon compound which are bonded by silt and clay particles, we say this is chemical stabilization, and the term is mineral associated carbon. And this mineral associated carbon was highest under tropical forest. You can see it's more than 75% tropical forest. This is quite interesting compared to the temperate or boreal forest. And more mineral associated carbon means that is more protected and cannot be easily oxidized. If you get time, you can study this paper by Kate Hickman in Global Change Biology. And they also studied the signature of 14C through different depth, and with depth they found a significant increase in persistence of carbon under different particularly free particulate occluded as well as mineral associated organic carbon. And that means what we studied in CSSRI with depth, the passive pool carbon content increase, though it is through a different method, and it is proved by their study. They studied 14C isotopic signature for that, and they also found significantly higher persistence at lower depth. And this is, you can see, this is Casuarina equisetifolia in Orissa coast near Chilka Lake. You can see the left side, the amount of leaf litter on the surface is around 3 to 5 centimeter, 3 to 5 centimeter leaf litter depth. And after decomposition, this will go to the lower depth. And particularly here, the soil is sand content is higher. So after decomposition, most of the carbon coming from the leaf litter possibly go to the lower depth, and there will be more stabilization. But the stabilization, I'm not sure because the sand content is higher. So how much will be the chemical stabilization? It will be interesting. And this Casuarina equisetifolia, these trees are used as shelter belt in those near the sea coast of Odisha. And now I was talking about the plant derived. They studied Heckman et al. In GCB. The microbial derived carbon and plant derived carbon. Microbial derived, this is the comparison between rice and maize system. We know whenever any residue we add from outside in the form of FYM or the plant input through roots, through isodeposition, this contains lignin, cellulose, unicellulose, and the microorganism starts working on that. So after the death of the microorganisms, we say this is necromass. We mostly measure microbial biomass, that is the living biomass. But this is the dead microbial, dead biomass. So this is called necromass. That necromass carbon is very much efficient in bonding with silt and clay particles. So this is called microbial derived carbon. And the undecomposed material that cannot be decomposed by microorganisms, that is, we say this is plant derived carbon, lignin, cellulose, unicellulose, those things. And under waterlogged rice condition, this plant derived carbon is more because the microbial activity is less under anoxic condition. But in upland condition, particularly maize, and whenever you go for agroforestry systems in arable soils, the microbial derived carbon was much higher. That is, the necromass carbon was much higher in agroforestry system. and that is more stable, cannot be oxidized. So in that way we can say the agroforestry systems are efficient, or maybe efficient, probably they are efficient in sequestering more carbon in stable forms. So there are methods for measuring microbial derived carbon as well as plant derived carbon. If the facilities are available, you can measure those fractions, and you can say that your system or agroforestry system is efficient in sequestering more carbon, stable carbon, compared to the agriculture systems, cereal-based systems. Now, coming to another example, root water uptake by the tree species is very important, and it significantly influence the below ground plant. BGBC means below ground biomass carbon. And what I was talking about, carbon use efficiency, here CUE is carbon use efficiency. Carbon use efficiency means like nutrient use efficiency, how much carbon is going inside the microbial cell, that is called carbon use efficiency. Through anabolism, we want more anabolism, less catabolism. So more carbon use efficiency means more microbial biomass, and MNC is the microbial necromass carbon. So more microbial biomass means more microbial necromass carbon, and this necromass carbon has entombing effect to soil organic carbon pool. Entombing effect means the efficiency of the microbial cells to take up more carbon inside the microbial cell. This is called entombing effect, and this ultimately result to contribute to soil organic carbon pool through in vivo turnover. And recently there is a study that I was talking about a few slides back. The plant derived carbon is called particulate organic matter, and microbial derived carbon, which is bonded to the silt and clay, is called mineral associated organic carbon. So this POM and MOM was significantly varied under different land uses, and it has been found that about pasture system, mineral associated organic carbon was significantly higher compared to the particulate organic matter carbon, as well as under compared to different cereal grain systems. So you can imagine the importance of pasture or grassland, as well as with agroforestry systems. There should be, or maybe in India we don't have much data on that. Maybe you will all study that and can report. It has higher efficiency. Agroforestry or grassland system has higher efficiency in storing more and more carbon in mineral associated organic matter, provided the soil has quite a higher amount of clay, maybe 15 to 20 or 30% clay on silt. Otherwise, like in soils of Rajasthan where sand content is higher, there may not be a higher amount of mineral associated organic carbon, but for soils like insecti soils or NP soils of UP, or as well as in central and western India, in southern India where there are black soils, surely and definitely there will be higher amount of mineral associated organic carbon, and this carbon is more stable compared to the other forms of carbon. Now, till now, we don't know at what point this microbial necromass or the microbial derived carbon is converted to soil organic matter. The most important point is whatever organic substances we are adding from outside, as well as the input from plants through roots, that organic substances, we know the end point, but at what point this plant derived or external carbon input is converted to soil organic carbon, we don't know, or soil organic matter, we don't know, because this conversion of external sources or plant carbon to soil organic carbon or soil organic matter is very important. More conversion we need, and that at what point the conversion, we are measuring the end product as well as before the system, both ends, what we are getting, we are measuring that. But where is the transition point? At what point this microbial derived carbon or soil plant derived carbon is converted to soil organic matter or soil organic carbon? We don't know. So this is one of the researchable issue what globally people are going after. Now this is one of the land levels at CSSRI, Eucalyptus teritigonis. We collected the leaf litter. during the last 1 year, and we found the weight was 1 hectare about 4.66 ton per hectare. It contains about 35 to 36% of carbon, and if we calculate that, it's about 1.66 ton carbon per hectare per year. This is only leaf litter. It also includes the twigs, but not bark. Bark fall is also happened under eucalyptus trees. So just imagine in 1 year, we are adding only through leaves about 1.66 ton carbon per hectare to the soil. And how much carbon is going to soil through roots, which are formed through photosynthesis? We don't know. We don't have data on that. Obviously, it depends on the above ground biomass. Maybe the leaf, if the number of leaves are higher, obviously the photosynthesis will be higher and more carbon will go to soil through roots. As the roots, root biomass is also important in that case. So only leaf litter, we are getting about 1.66 ton carbon. So just imagine how much carbon will be going to soil through roots. So it's we don't have data on that. You can study those participants if they have facility. They can study that. Through allometric equation, you can calculate the above ground biomass. Carbon is there, how much there. But how much carbon is going to soil through roots? There are also some calculations, some equations are there for cereals, but for tree crops, I don't know. There may be something. This is the leaf litter collection trap what we used in our study, and we also studied the decomposition of different leaf litter. We placed different leaf litter. This is under Imblica officinalis. Away from the roots, away from the trunk of the tree, we put different places the leaf litter, and we studied the decomposition of those leaf litters. And we observed the significant difference. Means the decomposition of leaf litter at different places, different lengths away from the trunk varied. It's not similar in all the places, and it also depends on the that means the tree or whatever land use you are studying has significant effect, and it varies from trunk to different lengths from the trunk. Similarly, we also studied, we also collected the litter fall of jamun, CGM cumini, and it also contains, the leaf litter also contains about 35% of carbon, total carbon, and we calculated that it's about 4.61 ton per hectare. So you can see here the above ground biomass is higher than the eucalyptus, and also the number of leaves must be higher compared to the eucalyptus. So the contribution of that carbon, the leaf carbon to the roots for jamun should be higher. So we don't know that how much carbon is going to soil through roots or rhizodeposition, those things. These are some of the researchable issues. And you can remember the earlier I showed the soil carbon distribution under jamun plantation and eucalyptus plantation. The passive pool carbon was higher below 1 meter soil depth under jamun. So from roots, the amount of carbon which is secreted by the roots of the jamun tree, the composition is different. That means the polyphenol content might be higher, and also the microbial activity is lower compared to the surface soil at below 1 meter soil depth. So whatever carbon is going to soil below 1 meter soil depth, that cannot be easily oxidized, and probably with the bonding with these soils are alphisols. So we found here silicate clay in each horizon at surface layer, but similarly at lower depth also about 10% clay we found. So that clay might have attached with that organic substances and formed stable complex, and microorganisms cannot able to degrade that. So that's why that might be one of the reasons for that higher amount of passive pool carbon on the jamun land. And that workshop recently, yesterday it was concluded. I supplied to Arbind some interesting observation, some interesting study they have reported. That is one of the, I was so surprised what we are not studying that. This, what they are talking about, canopy soils. Canopy soils means the soils are formed above ground biomass, means in tropical rainforest, particularly in west coast of India, Kerala, where there is a high rainfall, in the trees you can find so many epiphytes, and these epiphytes, through their roots, they also form roots, and you can see. I took photo of their slides. You can see there. This is really a new aspect, and I found. I must inform all of you. And they are studying in Costa Rica. In Costa Rica, the trees, above the trees, there are so many epiphytes, and the roots of the epiphytes are dependent on that tree. And after the decomposition of those roots, there is the formation of soil. And they are studying that soils. And how much carbon is there, how much, I was so surprised. I asked Professor Smith, Patton Smith, she delivered that lecture. And we know in 5 factors of soil formation, parent material is one of the important factors. And here, above the tree, inside the tree trunks or tree branches, from where you are getting parent material here. So what she replied, in tropical forest near pond or somewhere, there are rainfall, so there are dust particles in the winds. So that dust particles are precipitated on the branches, and from there the roots of the epiphytes are binding together to those dust particles, and by this way the soils are formed. And recently the USDA has transformed the definition of soil, and these soils comes under histosol soil order. And this is the paper, probably this year it will be published. You can all study that paper, this canopy soil abundance. And you can see the tree. In our place, so I am from West Bengal, northern part of West Bengal, North Bengal. We have also about 2,500 millimeter rainfall, annual rainfall. And these trees, you can see these are the epiphytes. So many also in our places, the old trees, they have this kind of epiphytes. And we did not notice that. So after the death of these epiphytes, sometimes mosses are also there. There is a formation of soil. So this is a new era of science. Really, it's 21st century, and we can report that also. And but it is very difficult to collect. You need to climb up the tree and collect these soils. In particularly in high rainfall areas, you can find in Kerala or Maharashtra coast, Western Ghats. And this is her result. What they found, about 8 times more soil organic carbon in canopy soils compared to the ground soils under that tree species. It's about 453 gram per kg compared to the ground soil about 57 gram per kg. So it's quite astonishing. And in terms of active carbon pool, it is also about 3 times higher active carbon, 3.53 gram per kg compared to the ground soil, 1.23 gram per kg. You can see these are the areas where canopy soils are mostly found. She also worked in India long back in Tamil Nadu. And you can see the west coast, the Western Ghats, due to high rainfall, these canopy soils are predominant. I did not have idea before that. So maybe whenever you visit Kerala and some places, you can see that. So in terms of conclusion, these are some of the conclusion. I am not going in detail. But the thing is, whenever you study, some of the precautions or some of the do's and don'ts, what we need to keep in our mind. Number one, for tree species, suppose in agroforestry system, in between 2 rows of trees, in between there is a cereal crops, maybe wheat or mustard, something. So while reporting how to take soil sample from those places, this is one of the main important issues. So you need to take samples separately from the cereals, maybe wheat, and the tree canopy, how much it spread from the tree, maybe poplar or something, else trees. You need to take sample from away from the roots within the canopy. Maybe it will fall under the cereal crop over the wheat also. So while reporting, you need to be very important, very cautious, because the roots of the trees goes much below 50 centimeters soil depth. So you need to, there is a necessary of, you need to take sample up to 1 meter depth, minimum 1 meter depth from the tree species. And for cereals, 30 centimeters is sufficient. And while reporting, you need to report that. And the second point is you need to have the bulk density data. In absence of bulk density, you cannot calculate soil carbon stock. And for reporting soil carbon data, in stock is very important. Only concentration in gram per kg or percentage is not sufficient. This is the globally accepted unit, carbon stock. You need, so for that reason, you need bulk density data. And through different depths, you can study the aggregate associated carbon or aggregates, those things. And without doing aggregates, you can also study the particulate organic carbon as well as mineral associated organic carbon. If some of you have experience of Yoder apparatus, for that, with that we measure soil aggregates. The last sieve, that is a 0.053 millimeter sieve, you take the 0.053 millimeter sieve and take some soil and sieve that in a tray in the soil. And above the sieve, some amount of soil will be there, and below the sieve in the tray, the soils will be there. So above the 0.053 millimeter sieve, collect the soil separately, and in the tray, what amount of soil is there, collects it separately. Above the sieve, the dry that soils, that above the sieve, we say just measure the carbon concentration of the soil. This is particulate organic carbon. That means it indicates particulate organic matter. Below the sieve, in tray, the liquid soil sample, dry that. This is the silt and clay. Silt and if you measure the carbon content of that, this is the silt and clay associated carbon or mineral associated organic carbon, what I was talking. So these two is very important. Up to different depth you can do. And other things, the plant derived carbon and microbial derived carbon, you need gas chromatograph for that. So this is difficult. So these 2-3 points are very important. You can follow this, and in your laboratory facilities, using that you can measure that only by using water black oxidizable carbon method. And for UP especially, there are abundance of inorganic carbon. I have not told much about inorganic carbon, mostly in terms of calcium carbonate deposition. You can also measure the inorganic carbon by measuring calcium carbonate. There are 2-3 methods and easy methods. Number one is first is rapid titration. Take the soil sample after sieving and using phenolphthalein and HCl. We need to add HCl on that, 0.5 normal HCl, and then titration with acid using phenolphthalein indicator. There are also methods. Calcimeter is also there for measurement of inorganic carbon. We have in our institute one calcimeter, 2 calcimeters. So after measuring inorganic carbon, you just if you measure total organic carbon or total carbon, subtract that from that, you will get total organic carbon. So with depth, inorganic carbon concentration will also increase, and report separately soil organic carbon as well as inorganic carbon. Because in tree land uses or agroforestry land uses, as the root goes much deeper, these calcium carbonates get dissoluted due to the root secretions. Acids are secreted, weak acids, and these acids helps in dissolution of those calcium carbonate concretions and nodules. And simultaneously, this calcium, after goes to soil solution through cation bridging, it also helps in organic carbon stabilization in lower depths. In whatever result data I have shown, there dissolution of calcium carbonate has played an important role, though I have not shown the inorganic carbon data. But in UP particularly, UP is the largest state where higher amount of more than I think 2 million hectare, about 2 million hectare alkali soil or sodic soils are there. So tree land uses or agroforestry systems are efficient in dissoluting those calcium carbonates and reclamation of natural reclamation, you can say, of those alkali or calcareous soils in Uttar Pradesh. Thank you. Any more questions? Questions, welcome. You can ask anything.
Thank you. Thank you very much, sir. It was really an enriching session. And I would give to the floor to the participants if they have any question, because after that, I also have a few questions. So participants, if you have any question, please, you may ask it now.
Good morning, sir.
Yeah, Dr. Himshikha, please.
Good morning, sir. My question is that can lignification, which happens in large trees, can it be a parameter to determine the carbon stock in any tree?
Are you talking about above ground biomass or soil carbon?
Sir, that means that lignification, because it is also in another way denotes the carbon accumulation in a tree.
Actually, I told you above ground biomass. You can calculate the total carbon stock in by that formula or through allometric equations, particularly using DBH. Are you from forestry background?
Ji, sir. Ji, sir.
Yeah.
Yes, sir.
DBH and plant height. That is sufficient. And after that, above ground biomass, what is happening is not. Being a soil scientist, I'm not telling you it's not important.
Fine, sir. Fine.
But that residues, whenever it goes to soil, that plant-derived carbon, lignin is very important component. Okay.
That's my point, is sir.
Yeah, and that lignin, after microorganisms starts working, some amount of lignin is converted to after a long time, because it's not easily degradable. It forms, it goes inside the microbial cell, and there is a death of the microorganisms. So we say this is necromass. Okay, and the lignin, what is left, this is called plant-derived carbon, and this plant-derived carbon, it is not as much stable compared to the microbial-derived carbon, but it is stable. So if higher amount of lignin is found in the soil, and not only lignin, there are so many lignin derivatives. Whenever we have a method also, if you want, I can send Dr. Arbind. You can study if you have chromatograph, gas chromatograph. That lignin also you can estimate in soil, not plant. And that lignin, that is, we say plant-derived carbon, if it is more, that carbon is more stable. And we want more and more lignin or some plant-derived carbon should come in the soil and go lower depth, so that it can be more stable. So I'm telling you, as you are from forestry science, you can see in India, mostly forestry people are mostly interested in above ground biomass.
Fine, that's it.
Yeah, and I have shown in the first slide, it contains throughout the biosphere about 560 petagram, and the soil carbon contains 2,500 petagram. So just imagine how important is the soil. So, and there are very less reports. Even we have our forestry scientists, agroforestry scientists in our institute. They are mostly interested in above ground biomass, less studied, and there are so many things to study in soil. So being a soil scientist, I know these things we should know, and maybe in future endeavor you can study those things.
Yes, sir.
I'll
Thank
send
you
you
so
the
much.
methods.
Fine, sir. Thank you so much, sir.
And these things, if you report those things, no, it will add your value. And while publishing nowadays, is very difficult to publish in high impact factor journal. And if you add this data, it will add value to your paper, and it will have maximum chances to get published. Otherwise, if you report only above ground biomass, simply they will say this is incomplete study. You need to report also the soil carbon, how much it is going on.
Yes, sir.
Okay.
Thank you so much, sir. Thank you so much, sir.
There's one more. Dr. Shama Parveen has raised her hand, but before that, Dr. Joseph has asked Can anyone share contact number of Dr. Ashim Datta? Sir, would you like to share your contact number, or shall we share your email ID with the participants?
I know Dr. Joseph. He's from
Okay.
Manipur, I think. We did. In this
Yeah,
case-
yeah, it's me, Asim. How are you?
Yeah. Fine. How are you?
I'm good. I'm good.
After a long time.
Yes, yes, yes.
How is
Nice
family
to-
there?
Yeah, I'm at CIV Imphal right now.
Very good.
Okay,
I have
okay.
shared my
so-
phone number. Okay.
Okay, thank you very much, Asim.
Great, great. That's good. Dr. Shama Paveen, yeah, please go ahead and ask your question.
Good morning, sir, and good morning to everyone present here.
Yeah.
Sir, my question was regarding the method of sampling adopted in a research farm, what we are having in our university. What is the exact method of sampling while we go at the farm for like assessment of this carbon sequestration?
You can see this. This is the Jamun orchard. Okay.
Yes, sir.
So what we did, or what we follow, in between two rows, inside the canopy, we mostly take soil sample in between two, and for collection of BD through auger it is not sufficient. You can only take sample of only soil sample.
Yes.
We need to dig a big profile here, and it's very tough, because we also, as a soil scientist, we also study the soil profiles. Otherwise, you cannot take BD. So in between two rows where canopy is there, not outside the canopy, means where leaf falls are occurring, there from this area only you need to dig a hole, and so that you can also collect bulk density also. So otherwise, without bulk density, if you collect only through auger, you can also collect from auger sampling within the rows. There are studies also from trunk to different length of maybe 30 centimeter, 50 centimeter, and then 100 centimeter, and with depth. You can also compare also, because there are very, in India particularly, there are very less study on those aspects. How much carbon concentration varied from, soil carbon concentration varied from length from the trunk. You can also study up to 30 centimeter, 50 centimeter, and then 100 centimeter within the canopy, and in the depth up to 2 meter, minimum 2 meter, how soil carbon can vary. Because all the tree species, they are different in their character, their root character. Some roots go deeper, some roots spread over the surface. So it depends on the tree species also. For Jamun, there are very less amount of most of the roots go lower depth, not over the surface. So we took here within the canopy, we dig about 2 meter soil depth, and from there we collected depth-wise soil sample with bulk density, with core sampler. With the core also, we can collect the bulk density data. And thus report, I again request all of you, report the soil carbon data in megagram or ton per hectare by multiplying the concentration to bulk density and soil depth.
Thank you so much, sir. There's one little doubt also regarding the above ground biomass. Should we go for the same method of sampling of trees?
No, ma'am, actually, there are, I'm not a forestry specialist, but there are allometric equations for different tree species. If you want, we have a principal scientist on forestry. I can send you the allometric equations. You are also from forestry, I think?
Yes, sir, I'm from agroforestry, yeah. Forestry.
So you need dBH and plant height. From there, you calculate. Those allometric equations are developed by researchers for a particular tree species. The equations are used. Just you need to measure the dBH and the height by through altimeter or something. There are some instruments also, but it does not. Destructive soil sampling also some researchers follow, but it's very cumbersome for big trees. And you can also develop allometric equations for particular tree species.
Okay, thank you so much, sir.
Thank you.
All right, so if any participant has any question, we still have a minute or two. So Dr. Yumnam is asking, sir, sampling should be done till how much depth? Going till 1 meter depth is necessary?
If the tree species, it depends on the tree species. You may have idea about how much depth the roots can go of that particular tree species. But for forest trees, for this jamun or eucalyptus, mostly 2 meters should be followed, because whenever you report this jamun, what we found, even below 2 meters soil depth also we found roots. It depends on the age of the tree. This is about 30 years of age of this tree. So if you have some idea about the root length of that tree, you need to have the data of that depth. Otherwise, whenever you publish a paper and it goes to a review, reviewer will ask you why you did not collect soil sample up to this much. It will be an incomplete reporting.
So that means we should have some prior knowledge how deepen the root could go.
Yeah.
There is no standardized protocol what should be the depth,
Yeah,
right?
mostly if the published literature in India, if you study, mostly 0 to 50, 15 to 30. For field crops, it is fine, no problem. But for tree crops, where the roots goes much deeper than 1 meter, report that you need to study the soil also, because it has for soil carbon study, because now the present thrust is more and more carbon you want to get down the soil depth, because it cannot be easily oxidized. So that way, otherwise it will be difficult to get published.
Okay. So before I go to Dr. Amarjeet, there is a question before him, by Dr. Abha. She is asking, I would like to know about the carbon sequestration potential in mycorrhiza.
I'm not a microbiologist, but anyway, mycorrhiza has a very important role, because recent study in globally what they are reporting, it's very important, because the fungi. One thing I did not tell here, the fungi is very efficient in converting plant-derived carbon to microbial-derived carbon, because fungal biomass is much higher. That workshop also, what I showed you, Dr. Professor Karsten Muller, his topic was that on the microbial-derived carbon, and fungi has played an important role. And in fungi, the mycorrhiza is very important, arbuscular mycorrhizal fungi, and you need to study that those things. But I also asked him, because fungi are mostly dominant in acidic soils, but for alkali soils, sodic soils in Haryana or Western UP, what can happen? He also didn't have the answer of that. We need to study that. You can study that. You can report, because we don't have any data on that.
Fair enough, sir. Dr. Amarjeet, please ask your question. Dr. Amarjeet, you have raised, yeah. Dr. Amarjeet, we can't hear you. Please unmute yourself and then.
Yeah, now I am audible.
Yes, yes, sir.
Yeah, you are audible now. Please.
The two things are in my mind. One is that is temperature. We are in temperate areas. The temperature is having any role on the availability of the soil organic carbon, because the temperature plays an important role in the decomposition of the leaf litters and other things. One thing. Second thing is that adopting the agroforestry. Second thing is that the adopting agroforestry, we are having the very much one component of canopy management. When we manage the canopy of the tree component, then there is, can we say that there is a these availability of these litters and availability of these leaves into the soil carbon, for adding the soil carbon? Please, sir.
Yeah, temperature has very important role. I have seen I have shown one of the slides. We have studied the decomposition of different leaves. You can see in the left side within the bricks, the decomposition of the leaf litter. And in our tropical and subtropical countries, and due to high temperature in northwest India, more than in summer we have more than 45 degrees Celsius, and there is a fast decomposition. If there is a decomposition is necessary for supplying of nutrients. But that one slide I have shown, there must be more anabolism than catabolism. If there is a more decomposition, more CO2 emission will be higher, and microbial inside the microbial cells there will be less amount of carbon. So after death, the necromus in necromus there will be lower amount of carbon. So the conversion of leaf litter carbon to soil organic carbon, you need to study. We need to study. We don't have data on that. How much that we have collected the litter? About 4.66 ton, about 1.66 ton carbon per hectare. We are adding through leaf litter. How much of that 1.66 ton carbon is going to soil organic carbon? We don't have that data. We need to study that. And this is very important. But for tree species, this amount of leaf litter is very low, is very less compared to the contribution to roots through rhizospores and those things. So there are so many researchable issues on that, and agroforestry scientists as well as forestry scientists, soil scientists should be there to study all those things, to unveil all those things. So agroforestry, forestry, they have a very good scope to study so many things if someone is interested. And canopy management, that's what I told is also important, because in different away from the trunk, the decomposition rate is different compared to the near the trunk. So there are so many things, and also it depends on the soil type, what type of soil is there. Not only temperature is one of the factors, but soil type, also the management, irrigation, if you have irrigation, those things. So at what interval you are applying irrigation. So there are so many things, so many factors you need to consider while studying those things. Thank you.
Thank you, sir.
All right. May I ask a question, sir?
Yes.
I'm the course director, and I do have a question here. So when you were talking about the canopy soil, you also touched upon the biodiversity. So do we have substantial data to prove that canopy soil contribute in enriching the biodiversity? And the second part of my question is, are there any studies which can correlate the rich canopy soil could contribute in enriching the wildlife as well?
Actually, Dr. Smith is working on Costa Rica. You know, in Costa Rica is a tropical rainforest. This whole Costa Rica rainfall is more than maybe I think 2,300-3,000 millimeter per year. So these things she did not tell in detail. I also day before yesterday came to know about these canopy soils. So maybe in India, in Kerala coast, as you are from forestry, maybe you can contact those scientists whether they are working or not. But this is very interesting. So many things. What according to her, so many things are there. So she also, after a few years, maybe if this paper gets published, and then also you can know. I also don't have idea on that. Maybe.
Okay, sure. No problem. So that was really fascinating, and being a wildlifer, I also got found it really interesting, which bound me to watch your presentation and understand the preliminary things that I could grab from a presentation. We really thank you, sir. And for a formal vote of thanks, I would now invite Dr. Arbind Kumar Gupta to deliver a formal vote of thanks to you. Dr. Gupta.
Thank you, sir. Thank you, sir. Very good morning, everyone. Sir, you have your presentation is very informative and very interactive. On behalf of the organizing committee, I would like to sincere gratitude to our speaker, Dr. Rasham Gupta, sir. You have covered the integral part of natural resource, that is soil carbon, and you cover the soil carbon sequestration and their mechanism, agroforestry system practices for management of soil carbon stocks, carbon distribution in different depth of soil under agroforestry system, the impact of forest types on soil carbon, carbon use efficiency and how it can be managed or how it can be enhanced, and one more thing, that is canopy soil. So with these words, I would like to thank Dr. Asim Dutta, sir. Once again, thank you, sir.
Thank you all.
Yeah, thank you. Thank you very much, sir.
Bye. I have to go. Bye.
So you may, yeah, bye. You may please sign out, sir. We have a small announcement for the participants. Dear participants, it was really an interactive session where you played an instrumental role by interacting and asking your questions. We sincerely hope that it will continue. Just a small thing, I would ask to unmute Galaxy M31. We are not identify you, sir or ma'am. Either you please rename yourself, or otherwise we won't be able to give you mark your attendance. Galaxy M31. There is an account, a participant has joined on the name Galaxy M31. Anyway, I will exclude him or her from the next session. So the next session will resume at 11:30 as per the schedule. So we can take a 15 minutes break before the next session. Thank you. Good morning, participants. Welcome back after a 15 minutes break. Hope you have all refreshed and looking forward to the next lecture. Let me first formally welcome our next resource person, Dr. RLS Sikarwar. Welcome, sir. Welcome to the Forest Management for Sustainable Use of Natural Resources, a 21 days virtual training program organized by the College of Forestry at the Banda University of Agriculture and Technology. It is a privilege to have you, sir, here. And on behalf of the organizing committee and my own behalf, I welcome you once again. Today is the second day where, under the theme of natural resource management, planning, conservation, and reclamation, we welcome you. And dear participants, as Dr. RLS Sikarwar is an authority in the medicinal plants, and I remember in the yesterday's introduction and interactive session, most of you expressed your keen interest in the medicinal plants. So this is one of the most important lectures we have. And let me formally introduce Dr. RLS Sikarwar. He did his MSc, MPhil, and PhD in botany from School of Studies in Botany from Jivaji University in Gwalior. Thereafter, he joined CSIR, National Botanical Research Institute in Lucknow from 1990 to 2003 under the supervision of Botany Authority, I would say, in India, Dr. J.K. Maheshwari and Dr. S.K. Jain. After that, Dr. Sikarwar joined the Arogya Dham, Deen Dayal Research Institute, Jan Shikshan Sansthan in Chitrakoot from 2003 to 2020, very recently. He has published almost 7 books and over 230 research papers. So congratulations to Dr. Sikarwar for the double century. Papers in the national and international journals and recipients of several prestigious medals and awards, such as Dr. S.K. Jain Medal 2004, Gyan Chandra Medal in 2010, B.N. Malhotra Medal in 2016, and Pushpagyan Biodiversity Access and Benefit Sharing Award in 2020 for his incredible contribution in the field of taxonomy, ethnobotany, medicinal plants, and biodiversity conservation. At present, Dr. Sikarwar is working as Director, Center for Traditional Knowledge Research and Application and Professor at the Department of Environmental Sciences at AKS University in Satna. So we look forward to you, sir, and with a formal welcome, I invite you to kindly deliver your lecture. Over to you, sir.
Thank you very much, sir. Slide दिख रही है बढ़िया.
Yes, sir. Yes, sir. Ekdam, sir. You are visible along with your presentation, sir. Please.
Okay. Thank you very much, sir, for nice introduction. Dear participants and organizers, my lecture is on overview of medicinal plants status and conservation in India. So the relationship between man and plants is as old as human civilization. The plants provided 3 vital basic needs since ancient times, that is food, cloth, and shelter. The 4th most important basic need is medicine, which is provided by the plants and used by man since thousands of years. Mentions of medicinal plants, virtues of hundreds of plants in India has been made in the ancient works. There are hundreds of medicine plants mentioned in our ancient literature like Rig Veda, Atharva Veda, Charak Samhita, Susru Samhita, and Ashtanga Hridaya, etc. As per WHO, 50,000 species of plants as used as medicine in the world. Out of 50,000, 8,000 species used in India. So medicine plants mentioned in the literature, Rig Veda 67, Yajur Veda 82, Atharva Veda 288, Upanishad 31, Charak Samhita 620, Susur Samhita 775, and 910 in Ashtanga Hridaya. Besides this, more than 300 plants mentioned in the 18 Puranas, and 200 in Valmiki Ramayana, 59 in Ramcharitmanas, and over 100 plants mentioned in the Mahabharata. So what is medicinal plants? Actually, there is a one shlok, Mantram Aksharam Nasti Nasti Moolam Anosadam Ayogyo Purso Nasti Yojak Tatra Durlabh. कोई ऐसा अक्षर नहीं जिससे मंत्र न बनता हो, ऐसा कोई पौधा नहीं जिससे औषधि न बनती हो, ऐसा कोई पुरुष नहीं जिसमें योग्यता न हो। कमी है तो पारखियों की कमी है कि उनके गुणों को ढूंढ सके। तो every plants found in this universe has medicinal value or useful to the man in any kind. So medicinal plants, for formally definition, a medicine plant is a plant that is used to maintain health, to be administered or for a specific condition, or both, whether in modern medicine or in traditional medicine. Food and Agriculture Organization estimated over 50,000 medicinal plants are used across the world. A new study released by Kew Royal Botanical Garden in London, 28,107 plant species are being used medicinally, mostly in the non-industrialized world. WHO estimated about 80% of the population in the developing world rely upon medicinal plants for their primary healthcare needs. So maximum 90% population of Ethiopia use medicine, dependent on the medicine plants for their treatment of ailments and diseases. India 70%, Rwanda 70%, Tanzania 60%, and Uganda 60%. So there are several traditional systems are prevalent in different parts of the world. The traditional medicine systems, including the traditional European medicine, traditional Chinese medicine, traditional Korean medicine, traditional African medicine, Indian traditional system of medicine, including Ayurveda, Siddha, and Unani, ancient Iranian medicine, Islamic medicine, Muti and Ifa, scientific disciplines which study traditional medicine include herbalism. Herbalism is European medicine actually, ethnomedicine, ethnobotany, and medical anthropology. So these are the different systems which are prevalent in different parts of the world. Indigenous system of medicine. Indigenous system is a system which is apart from the allopathy medicine. So an indigenous system is a natural form of medicine outside the stream of Western or allopathy medicines practiced by majority of doctors all over the world. These systems are Ayurveda, Siddha, Unani, Yoga, and homeopathy. Ayush. It is Ayush now. It is Ministry of Ayush has been established in our country by the government of India. Ayurveda, Yoga, Unani, Siddha, and homeopathy. These are known as Ayush. In our country, there are two medical systems are prevalent. There are two streams are flowing in the form of traditional medicines in our country. One is documented and well codified system, जो मतलब लिखित और एक संहिताबद्ध सिस्टम है, इसमें including Ayurveda, Unani, Siddha, Amchi, and homeopathy. So this system is well codified and well documented system. In this system, around 2,000 species are used as medicine in these 5 systems, Ayush system. But in another system, which is very important system, that is orally transmitted system, that is not codified or not documented system. So orally transmitted system is very strong stream in our country. This system includes tribal medicine, folklore medicine, or ethnobotany. Ethnobotany is a part of tribal medicine is a part of ethnobotany. So in this system, more than 8,000 plant species used as medicine, while in Ayush system only 2,000 species are used in medicine. But in folklore system, that is orally transmitted system, 8,000 species used as medicine in our country. So the medicinal plants used in different systems: ethnomedicine 8,000, Ayurveda 18, Siddha 800, Unani 700, and Amchi 600. In Ayurveda, all the systems of the codified system, 2,000 plants are mentioned. But during the Charak period, the Charak has mentioned in the Charak Samhita, औषधीनाम रूपाभ्याम जानते यज पावने अविपाश्यैव गोपाश्य ये चान्यवन वासिना. Yani the forest dwellers, cow grazing cow herds, sheep and goat grazing shepherds identify the plants found in the forest by name, form, and medicinal property. So students of Ayurveda should also get knowledge of medicinal herbs from them. यानी जो चरक संहिता में लिखा है कि वनों में रहने वाले आदिवासी, गाय चराने वाले ग्वाले, भेड़ और बकरी चराने वाले गड़रिये, ये पौधों को जंगल के पौधों को उनके नाम, रूप और गुण से पहचानते हैं. आयुर्वेद के लोगों को चाहिए कि उनसे भी ये ज्ञान प्राप्त करें. तो इट मीन्स दिस ओरली ट्रांसमिटेड सिस्टम वाज आल्सो प्रेवलेंट ड्यूरिंग द Charak period. The same thing mentioned in the Susur Samhita, Gopala Istapasa Vyadha ye chane vancharan hai mool harash ye te byo vesajam vyaktrisyate. Dhanvantari Nigantu mein bhi hai kirat Gopalak tapsada bane charast kuslas tatane vidati nana vidya bhesh gyana praman varan kram naam jati. तो ये मतलब ये जो सुश्रुत संहिता, चरक संहिता और धन्वंतरी निघंटु में जो लिखा है वो चरक और सुश्रुत के समय भी ओरली ट्रांसमिटेड मेडिसिनल सिस्टम था, वो प्रेवलेंट था. और इंडिया, द कंट्री, आवर कंट्री इज रिच इन बायोडाइवर्सिटी ड्यू टू डाइवर्सिफाइड क्लाइमेटिक कंडीशंस. All 15 types of climatic conditions of the world found in India. That is vary from humid tropical Western Ghat and the hot desert of Rajasthan to diversified northeast regions of the country, from cold deserts of Ladakh and the icy mountains of Himalaya to the warm coast of peninsular India and central fertile plains provide numerous microhabitats. So the India is only country in the world which has 6 ऋतुएं, seasons. यानी जो 6 ऋतु हैं वो किसी कंट्री में नहीं होती हमारे भारत को छोड़ करके. और जितनी भी climatic conditions हैं, all climatic conditions are found in our country. If you go to the Himalayan region, Leh or Ladakh, the European country, you can enjoy the atmosphere of European country. If you go to the northeast, you can enjoy the Latin American countries' climate. If you go to the Arabian country, you visit our Rajasthan Thar desert, you enjoy that conditions there. So every conditions of the world is found in our country. That is the beauty of our country, and that's why India is rich in biodiversity. This is phytodiversity of our country. This is the latest data released by BSI in 2020-21. So there are about 54,733 plant species has been documented or listed in our country. Out of which angiosperms 21,849, gymnosperms 82, pteridophytes 1,310, Browphyta 2,791, fungi 15,504, algae 8,979, lichen 2,961, and microorganisms including bacteria and virus 1,257. Total 54,733 plant species are found in our country. Out of the flowering plants, 21,849. Out of which endemic plants are 4,381. That is the endemic plants, which plants only found in our country, not found elsewhere in the world. That is called endemic plants. And India is rich in endemic plants. 4,381 plant species are endemic. That is not found anywhere in the world. Out of which the threatened plants are 2,704 plants are threatened, and medicinal plants are 8,000. This is our country is divided into 10 biogeographic zones: Trans Himalaya, West Himalaya, Indian Desert, semi-arid zone, Western Ghat, Lakshadweep Island, the Deccan Plateau, Northeast, Eastern Himalaya, and Gangetic Plain. So the distribution of medicinal plant, if you see the Trans Himalaya have 700 medicinal plant species, West Himalaya 1,700 plant species, Endo-Gangetic region 1,000 plant species, East Himalaya 1,200 species, Northeast 2,000 species, then semi-arid zone 1,000 species, Indian Desert 500 species, Deccan Plateau 3,000 species, Western Ghat 2,000, and Indian coast 1,000 species. So this is the distribution of medicinal plants according to the biogeographic zones. This is the Valley of Flower. The Valley of Flower National Park, it is the bouquet of nature. This valley discovered in 1931 when 3 British mountaineers led by Frank Smith. They were climbing on the Kamat Parbat, and after the successful expedition, they during the return, they lost their way and reached to the Valley of Flowers, and very astonished to see the enchanting beauty of this Valley of Flowers, and they came back to our base camp and written a book on Valley of Flowers. Since then, it came into existence of this botanical world, and now the government declared it this as a national park, and UNESCO as a World Heritage Site. Are more than 500 plant species, including medicinal and rare endemic plants found in this Valley of Flowers. This is the tribal status in India. The 705 tribal communities, 227 ethnic groups, distributed over 5,000 villages, constitute 10.4 million population, 8.6% of total population of India. These tribals are actual custodian of the traditions related to medicinal plants. There are about 116 different dialects and 2 to 7 subsidiary dialects spoken by the tribal of India. They reside in and around the forest area and utilize the forest resources for food, shelter, and medicine. So this is the beauty of our tribal culture. Actually, why I am telling about the tribal, because the study of the plants used by the tribals, this is called ethnobotany. So the term ethnobotany actually coined by Johann W. Hertzberger in 1895. But in our country, we are using plants since ancient times. The relationship between man and plants mentioned in our all the Indian scriptures, Veda, Purana, Mahabharata, Ramayana. So the term ethnobotany given by Hertzberger, but it is used by our country since last 100 or 1,000 years, or beginning of the civilization. So ethnobotany is the study of plants used by primitive aboriginal people. This definition given by John W. Herzberger, but now the plants and people's relationship is ethnobotany. So in India, ethnobotany started by E.K. Janaki Ammal, but it was nurtured by Dr. S.K. Jain, regarded as the father of Indian ethnobotany. So study of ethnobotany is the Ministry of Environment and Forest funded a mega project on documentation of ethnobotanical knowledge, All India Coordinated Project on Ethnobiology. Under this project, 25 centers have been made across the country, different zones, and all the documentation related to the tribal people have been documented. And out of this, when Dr. Pushpangadan compiled this information, the 10,000 species used by the tribal community in our country. So the 10,000 plants used by tribal community in different purposes, including edible plants, 4,000, fodder, 500, fiber and cordage, 600, essence and perfumes, 100, gum and resin, 300, other uses 750, pesticides and pesticides 325, and medicinal use 8,000. So this 8,000 species I have already mentioned. This is apart from the Ayurveda system. Breakup of life form of medicinal plants: tree 26%, shrubs 17%, herbs 41%, and climbers 16%. So the majority of the plants used as medicine is herbs, 41%. Herbs means most of the plants used as panchang, all the parts, root, fruit, stem, leaves, and fruit, flowers. So panchang used by all herbs, so that is 41%. That is why the medicine plants are depleting fast from from nature, because of over exploitation, because these includes roots, leaves, fruits, flowers, and stems. So panchang is used by herbaceous plants. That is why the plants are depleting. Then plants parts used as medicine. So root 30%, seeds 4%, fruits 7%, flower 10%, stem 6%, bark 14%, rhizome 4%, and wood 3%. Whole plant 16%. So whole plant and roots constitute 46%. This is the major cause of depletion of medicinal plant. Then distribution of medicinal plants in the dominant families. So Fabaceae is the largest family, having 117 plants as a medicine. Then followed by Euphorbiaceae 61, then Poaceae, Asteraceae. This is the degradation form. Then modern drugs developed from plants. So nowadays about 122 drugs have been derived from the plant sources, out of which 30 drugs developed in India, such as just artemisinin, anti-malaria drug developed from Artemisia annua. Then caffeine stimulant, Camellia sinensis, codeine, Papaver somniferum, Aldopa, Mucuna pruriens, केवाच इसको कहते हैं, डिजिटॉक्सिन, कार्डियोटोनिक, digitals, purpureas, ephedrine, gossypol. So these are the drugs developed from plant sources. Ephedrine, bronchodilator, ephedra, sinica, eugenol, sorelin, Jeevani, Trichobacillus. These are some important medicinal plants used in different organs. So this is for nervous system, the Echurus calamus, Bacopa moneri, and Convolvulus prostratus are used in nervous system. Then immune system, Tinospora cordifolia. So Tinospora cordifolia play a very significant role during the corona period. So it is immune system, it is used. Then digestive system, aloe vera, Phyllanthus amarus, and this Ipomia turbinata. So these are used in digestive disorders. Then likewise integument systems, neem, bakshi, and karanj. So these plants are different plants used in different organ systems, disorder of these organs. Respiratory system, जैसे cordifolia, sibilingi, and this kevach. Medicine plant used in different formulations. So Triphala constitute of Phyllanthus amli, Halobehida amla, Phyllanthus amlica, Terminalia cebula, and Terminalia balearica. So Triphala is used in 219 formulations, different formulations, Ayurvedic formulations. Then Glycyrrhiza glabra used in 141 formulations. Piper longum 135 formulations. Justicia adatuda 110 formulations. Bidania somnifera 109 formulations. Cyprus rotundus 102, Tinospora 88, and Berberis arrestat 65. So these are the major medicinal plants which are used in the maximum formulations for the treatment of ailment and diseases. There are currently 250,000 registered medical practitioners of Ayurveda system as compared to the 7 lakh of the modern medical system. Modern medical system, 7 lakh doctors are involved, but in Ayurveda system, 250,000 registered medical practitioners. In rural India, 70% of the population depend on the traditional type of medicine. So there are estimated to be around 25,000 effective formulations used in a folk medicine and known to rural communities all over the India. And around 1,000 design formulations are available in indigenous medical texts. So in the Ayurveda, 10,000 formulations are mentioned, but in our ethnomedicine, 25,000 effective formulations are used. Medicinal plants exported from India. So these are the plants which are exported from India. Aconitum, Acorus calamus, Justicia adatola, Casia angustifolia, Anula reshivosa, Picrohaja crua. Then medicine plant imported from India. These plants which are not plenty available in our country, so we have to export from other countries. Cascuta epithemum, Glycyrrhiza glabra, Lavandula, Stochius, Operculum terpethum, Pimpinella, Anisum, Smilex china, then Smilex ornata, or Thymus vulgaris. So why herbal medicine? Why herbal medicine? So because it is cheap. It is cheap, easily available in all seasons, easily prepared, no technology involved for preparation of formulations, then no side effect, utilized for 100 years. It is utilized by our forefathers 100 of years. So it have better compatibility to the human body. But it is mentioned in the Charak Sanghita, Yatha Visam, Yatha Sastram, Yatha Agniras Nirratha, Tadosatam, Vigyatam, Vigyatam, Mritam Yatha. Charak Sanghita में चरक कहते हैं कि हम लोग जो आज की डेट में कहते हैं कि हर्बल मेडिसिन्स है वो उसका कोई साइड इफेक्ट नहीं होता, लेकिन ऐसा चरक संहिता में ऐसा नहीं है। चरक संहिता में चरक ने लिखा है कि अगर आपको किसी पौधे के गुण धर्म के बारे में मालूम नहीं है तो वो यथा विषम यथा शस्त्रम यथा अग्निरस निर्यथा तदौषधम विज्ञातम विज्ञातम मृतम यथा, यानी अगर आपको उस पौधे के बारे में मालूम नहीं है तो वो विष के समान, शस्त्र के समान और अग्नि के समान हानिकारक है। और तदौसदम विज्ञातम विज्ञातम मृतम यथा, और अगर आपको उसकी गुण धर्म के बारे में मालूम है तो वो अमृत के समान गुणकारी है। तो हम जो हर्बल मेडिसिन है उसको ग्रहण करने से पहले उसके पूरे गुण धर्म देख लें, तभी उसका उपयोग करें। अगर गुण धर्म मालूम नहीं है, ऐसे ही खा लिया, तो वो विष और शस्त्र के समान या अग्नि के समान हानिकारक हो सकता है। सो दीज आर द सम endemic or rare plants also used as medicines. Then why not modern medicine? So because it is costly, beyond the reach of local people, not available in remote areas, causes side effects, expiry date. So these are the factors. That's why we should not prefer the modern medicine. या फिर एक श्लोक है, या हुदीर्ण समयंत नान व्याधिम करोति च सक्रिया नत व्याधिम हरण्य मुदीरेत। यानी जो चरक संहिता में लिखा है कि सही औषधि वो है जो रोग का शमन करे, यानी रोग का शमन करे, न कि वो जो एक रोग का तो शमन करे लेकिन दूसरी व्याधि उत्पन्न कर दे। यानी तो साइड इफेक्ट का वर्णन हमारे यहाँ पहले से ही है कि सही जो चिकित्सा पद्धति है वो पद्धति है, सही जो औषधि है वो औषधि है जो व्याधि का पूर्ण रूप से शमन करे, न कि वो पद्धति जो एक व्याधि को तो ठीक करे लेकिन दूसरे को पैदा करे। तो साइड इफेक्ट ऑफ मॉडर्न ड्रग्स किल मोर अमेरिकन्स एनुअली देन द वर्ल्ड वार सेकंड एंड वियतनाम वार कंबाइंड। यानी सेकंड वर्ल्ड वार और वियतनाम वार में जो जितने लोग मरे थे वो अमेरिका में साइड इफेक्ट से हर साल उतने लोग मर जाते हैं। Then loss of medicine plant diversity. According to the WHO, 200 species of medicine plants in India are endangered. Over 500,000 tons of dry raw material is indiscriminately collected from the forest every year. So these are some medicinal plants and also endemic and rare plants. So this is Electra chitrakutensis. It is endemic to our endemic to India. Now it is found in Madhya Pradesh and now reported from Rajasthan also. So, but it is very critically endangered and very highly medicinal plants. It is used as in leprosy. The anti-leprotic activity has been identified in this plant. So this is Sapria himalayana. It is a very rare plants, endemic plants of our country. It's a allied species of Rafflesia andoli. This is IUCN threatened plants, critically endangered 44, endangered 113, and vulnerable 87. So these are the plants. Saraka asoka is one of the most important plants of our country. Then Maconopsis aculeata, Renanthera scutiana, Venda cerurea, Rhizosphalia simpsoniana, Sapria himalayana. So causes of loss of medicine plants. So there are mainly two causes of medicine plants. One is anthropogenic, another is natural. So anthropogenic cause includes deforestation. 95% plants collected from forest, out of the 70% collected in destructive manner. Then loss of natural habitats by deforestation, expansion of agriculture, industries, construction of roads, railways, dams, then grazing, pollution. So these are the factors which are responsible for the causes of medicine plants, loss of medicine plants. Then nature is not behind. There are some causes include drought, floods, landslide, disease, lack of pollinator, invasion of alien weeds, the Parthenium, lantana, then Mecania. These are some other invasive species which are very dangerous to our indigenous biodiversity. So these are the, if tree is chopped, nuclear bomb is dropped. So these are the causes of loss of medicinal plants. Then there are 8 plant species which are mentioned in Ayurveda as Ashtavarg ki aushadhiyan. So Ashtavarg ki aushadhiyan including Jeevak, Rishabhak, Meda, Mahameda, Kakuli, Chir Kakuli, Riddhi or Vriddhi. So these are the 8 plants which are critically endangered. These plants used in our Chamanpras. So, but these are not available. So, in place of these plants, other substitutes have been used. That's why the Chamanpras is not so effective. Then endangered medicinal plants, Dolomia costus, Surya costus, Omipura vitae, Nardo stachys, Jatamasi, Rauwolfia serpentina. So these are all plants are critically endangered. Then loss of medicinal plants in our Bundelkhand region. So there are several high-value medicinal plants like Safed Musli, Chlorophytum tuberosum, Narkachu, Gingiber capitatum, Operculina petaloidia, Vidhara, Nirgundi, Kand, Electra chittugutensis, Ama haldi, Krupma amada, Isormul, Aristolochia indica, Gloriosa superba, then Eulopia herbaceae, Blaricum, Ureria picta, Pectinopteris radiata, then Helenia speciosa, Kyokan, Curculigo orchioides, Kali musli, Celestus paniculatus, Staculia urens, Chlorospermum religiosum, then Oroxylum indicum, Stereospermum suaveolens, and Emilia basal, etc. have been reduced to a critical state due to the over-exploitation and loss of natural habitats. This is Electra chittugutensis, the Helenia speciosa, this is Kyokan, one of the most important medicinal plants. Then Cordia maculoides, it is a tree, very critically endangered tree. Then Citrullus colocynthis, a Choti Indrayne. Gingiber gerumbat, Gingiber capitatum, Butea monosperm variety Lutea, Plumbago gelanica. These are the all medicinal plants. Then Eulopia herbaceae, Nervalia praniana, Tacca leontopetaloides, Operculina petaloidea. Operculina petaloidea is a real Vidhara, but instead of this, Argyria nervosa is used as Vidhara, but actual Vidhara mentioned in our the ancient Ayurveda literature is Operculina petaloidea. This is very, very critically endangered. Then Typhonium trilobatum, Aristolochia indica is one of the Isormul, one of the most important medicinal plants. Then Actinopteris radiata, Curcuma amada, this Rothika serrata, Bharangi जिसको कहते हैं, Ureria picta, Pitwan या Prashnopani, this is also endangered plants. Gloriosa superba is also depleting fast from the nature. Then Trichosanthis bracteata, it is also very rare. Actually, the majority of the medicinal plants found in the forest. So, in our Mahabharata, it is mentioned that Nasya Dwan Mrate Bragyan Bragyan Nasute Banen Banam Banam Rachat Vyage Brage Rachat Kananam. बाघों के बिना वन की रक्षा नहीं हो सकती तथा वन के बिना बाघ नहीं रह सकते क्योंकि बाघ वन की रक्षा करते हैं और वन बाघों की. इसी तरह से सिंह विहीनम विनम विनिश्चेत सिंहा विनश्ये श्रुतेन बनेन. सिंह से सूना हो जाने पर वन नष्ट हो जाता है और वन के बिना सिंह भी नष्ट हो जाते हैं. सो आज स्थिति ये है, जो लॉस ऑफ मेडिसिन प्लांट्स का जो कारण है वो ये है कि जंगलों में से शेर या बाघ खत्म हो गए तो लोगों का भय खत्म हो गया, तो पूरा जंगल खत्म हो गए. और जंगल खत्म होने से बाघ खत्म हो गए क्योंकि उनकी जो हैबिटेट है, टेरिटरी है, वो खत्म हो गई. तो मतलब वनों के संरक्षण के लिए बाघों का या सिंह का होना बहुत जरूरी है और सिंह होने के लिए जंगल का भी होना बहुत जरूरी है. तो आज परिस्थिति ऐसी है कि जंगल भी खत्म हो गए और शेर भी खत्म हो गए, जबकि हमारे पूर्वजों ने बहुत हजारों साल पहले ही इसके लिए आगाह किया था। Then story of Ayurveda. The story goes that Brahma ordered the sage Jivaka to found a plant or herb which has no medicinal property. Jivak wandered for 11 years in search of such a plant but could not find one. When he returned and informed Brahma of his failure, much to his surprise, Brahma recognized him as a great physician. यानी प्रत्येक जो पौधा है वो औषधि है। जब जीवक को ब्रह्मा जी ने ज्ञान आयुर्वेद की शिक्षा दी और परीक्षा लेने की घड़ी आई, तो ब्रह्मा जी ने कहा कि मुझे कोई ऐसा पौधा ला करके दिखाओ जिसकी कोई मेडिसिनल वैल्यू ना हो। और जीवक 11 साल जंगलों में भटकने के बाद बड़ा मायूस होकर के ब्रह्मा जी के पास पहुंचे, तो ब्रह्मा जी ने उसको अपने गले से लगा लिया कि तू मेरी परीक्षा में सफल हुआ। तो मतलब मंत्रम अक्षरम नास्ति नास्ति मूलम अनौषधम अयोग्यो पुरुषो नास्ति योजक तत्व दुर्लभ। ऐसा कोई अक्षर नहीं जो मंत्र न हो, ऐसा कोई पौधा नहीं जो औषधीन हो, ऐसा कोई पुरुष नहीं जिसमें योग्यता न हो। कमी है तो उनकी योजक की कमी है, पार्थियों की कमी है। इसी तरह से चरक भी कहते हैं नाना औषधम भूताम जगत किंचन, देयर इज नो मैटर इन दिस यूनिवर्स व्हिच हैज नो मेडिसिनल प्रॉपर्टी। किसी भी पदार्थ में या किसी भी पौधे में ऐसा नहीं है कोई न कोई इसमें मेडिसिनल प्रॉपर्टी न हो। Then conservation, the conservation of medicinal plants. So there are two ways of conservation, for everybody knows, for in situ conservation and ex situ conservation. So in situ conservation, Government of India established 106 national park, 564 wildlife sanctuary, 18 biosphere reserve, 7 world heritage sites, 46 Ramsar wetland, 218 community reserves, 99 conservation reserves. All these include 5.26% area of our country under protected area network. So these national parks having the conservation of medicinal plants, national park, wildlife sanctuary, etc. Then another way, removal of reproductive parts and conserved in seed banks, carrier banks, DNA banks, removal of whole plants from natural habitats and conserved in botanical gardens. So 131 botanical gardens are established in our country or found in our country. Then conservation measures, control of over exploitation. This should be stopped. Establishment of national park, wildlife sanctuary, and biosphere reserves. Cultivation of rare endangered certain plants species in the forest area. Establishment of botanical gardens should be established for ex situ conservation. Collection and preservation of materials such as fruits, seeds, pollen, and tissue. These banks should be established. Then legislation of conservation should be enacted. The legislation already, rules have been already enacted by Government of India, but the peoples are not following the instructions as given in the laws. Then extend a strategy for awareness to the local people. Then awareness will be increased in the mass communities. Motivation, training, exhibition, fair, handouts, plant species, print and electronic media should also be involved to the aware the local communities. Then why medicinal plants are not so effective nowadays? Because of adulteration and substitution of medicinal plants. So the adulteration and substitution. The adulteration is of two types: deliberately and deliberately. Means एक तो बिना जान पहचान, बिना जाने, और दूसरा होता है जान करके. तो जो अनडेलिबरेटली है, सेवरल प्लांट्स है, वन कॉमन नेम. एक ही पौधे, मतलब कई पौधों का एक ही नाम है. तो अगर किसी पंसारी के पास या किसी दुकानदार के पास जाओगे तो वो आपको जो फलाना मांगेगा वो दे देगा, उसको नहीं पता है कि हकीकत कौन सा है, क्योंकि वो नाम उसी का वही है. देन सेम प्लांट विद डिफरेंट वर्नाकुलर नेम. एक ही पौधे के कई नाम होने से कंफ्यूजन पैदा होता है. इसी तरह से डिफरेंट प्लांट्स विद सेम वर्नाकुलर नेम. लेकिन जो दूसरा है जो जानबूझ करके किया है, व्हेन ड्रग इज स्कार्स और कॉस्टली, जब कोई औषधि है वो बहुत कम है या उसकी महंगी ज्यादा है तो उसमें वो मिलावट कर देता है. जैसे काली मिर्च में पपीता के बीज मिला दिए, या काले जो मिर्ची में ईंट पीस करके मिला दी, तो मतलब वो जानबूझ करके कुछ न कुछ मिला देता है, उसको डेलिबरेटली अल्टरेशन कहते हैं। डिफरेंट बट सिमिलर लुकिंग मटेरियल अशोक एंड कुटज, अशोक की छाल की जगह पर कुटज की छाल का इस्तेमाल कर देते हैं क्योंकि दिखने में एक जैसी होती है। तो जो मिलावट है वो सबसे मेन कारण है कि मेडिसिन प्लांट्स की जो क्रेडिबिलिटी है कि रोग ठीक करने में मेडिसिन प्लांट ठीक नहीं करते हैं, उसके पीछे जो मुख्य कारण है वो कारण है उसमें मिलावट और सब्स्टिट्यूशन, यानी मिलावट और प्रतिस्थापन। और दूसरा भी जो हम आयुर्वेद पॉइंट ऑफ व्यू से देखें तो आयुर्वेद में लिखा है कि पौधों का जो कलेक्शन है वो बड़े विधि विधान से करना चाहिए, मतलब उनको आमंत्रित करके, नहा धो करके, और किस सीजन में किस दिन कौन से समय पर करना चाहिए। लेकिन आज की डेट में ऐसा नहीं, अभी ऐसे ही मतलब कहीं भी गए, कैसे भी गए, दिशा मैदान जा रहे हैं, लोटा ले करके उधर से उखाड़ लाए उसी हाथों से, तो ऐसे नहीं है। हमारी औषधियां हैं, ये दिव्य औषधियां हैं। आपको पता होगा जब हनुमान जी, लक्ष्मण जी मूर्छित पड़े थे, हनुमान जी बूटी लेने के लिए गए थे, तो वहां जो सारी औषधियां हैं वो हनुमान जी को देख करके लुप्त हो गई थी आज पर वाल्मीकि रामायण। तो हनुमान जी ने उनकी स्तुति की, और स्तुति की तो वो सारी प्रकट हो गई। तो हनुमान जी ने कहा कि इसमें से कौन सी संजीवनी है मैं पहचान नहीं सकता हूँ, और बाद में उन्होंने पूरा पहाड़ को उखाड़ लिया था। तो जो औषधियां हैं, जो औषधि पौधे हैं, वो बहुत दिव्य औषधियां हैं, दिव्यता है उनमें। तो हम उनको ऐसे ही अपने पैसे के कमाने के हिसाब से उनका मिसयूज करते हैं, इसी की वजह से उनमें इफेक्टिवनेस कम हो गई। देन अशोक रियल अशोक इज जेन्युइन अशोक इज सराका अशोका, सीता अशोक, but instead of Sita Ashok, Polyethia longifolia, Soria robusta and Bohania purpurea are used in place of Saraka Ashoka. Similarly, Bhui Amla, Phyllanthus amaris is genuine, but Phyllanthus fraternus, Madras pattern, urinaria and Davilis are used instead of Amarus. Then Sarva Hemidesmus indicus is genuine, but Cryptolopus buchanani, Ichnuca carpus frutescens, these are used in place of this. Likewise, Brahmi and Safed Musli, Borybelium is genuine, but Tuberosum and Arundinicium are used. So these are the, this is the Saraka Ashoka, real Sita Ashoka, but instead of this, we use this Polyethia longifolia. Now it is known as Monule longifolia. Then why correct identification? So one plant has 100 common names in different language and places. One common name of several plants. Several plants have one common name. So morphological similarity. So why the correct identification is compulsory for identifying a particular plant. So how to identify a plant? With the help of flora, country, state, district flora, regional flora available everywhere nowadays. With the help of herbarium, if nearby herbarium in your locality, you can identify your plant with the help of herbarium. The college, university, BSI circle, institutions, CNS. Then with the help of taxonomist, you can identify your material. Then identification key is provided in the flora for identification of particular species. So the identification of plant based on habit, trees, shrubs, climbers. So आपका प्लांट पौधा किस तरह का है, अगर वृक्ष है, श्रब है, क्लाइंबर्स है, हर्ब्स है, एनुअल है, बाइनिल है, पेरियनिल है, ट्यूबरस है, बल्बस है. First you have to identify on morphological basis. Then habitat, whether it is found in aquatic situations or terrestrial, marshy or slopes. Appearance, erect है, prostate है, twinar है, hairy है, glabrous है, rigid है, stout है, delicate है. So you have to think over that factor also. Then arrangement and types of leaves, one of the most identifying character. Types and color of flowers, types of fruits. That leaf shape and arrangements. There are different types of leaves: linear, ovate, elliptical, oblong, cordate, orbicular, sagittate, hastate, lyrate. तो आपका पौधे की पत्ती है वो किस प्रकार की है, वो आपको ध्यान में रखना है. इसी तरह से उसका मार्जिन कैसा है, सिरेट है, बेबी है, इंटायर है, डेंटेट है. सो देन एपेक्स एक्यूट है, एक्यूमिनेट है, ऑब्ट्यूस है, मेकुरिनेट है. किस तरह का है वो आपको देखना पड़ेगा. Here is Borrelia diffusa. So actual Borrelia diffusa is this, but instead of Borrelia diffusa, this Borrelia procumbens and this Portula castrum, I've forgotten the name. So actual base, but these are used instead of this. So if you identify these, the stem of this Borrelia, actual Borrelia diffusa is pink, and flower is also pink. But you see the stem is green and flower is white. Likewise, this Portula, I have forgotten the name. If you see this, this is entire different. The flowers of this Borrelia diffusa is come to the aerial portion of the inflorescence. But here this is found in the base of the leaves. So these are the differences you can identify easily from one plant to another plant. Likewise, the Sankuspi is Convolvulus prostratus. It is white flower prostate, but a volvulus alcinoid is used in place of this Convolvulus prostratus. So the flower is pink, blue. Like Brahmi, the Brahmi is Centella asiatica and Bacopa moneri. Both are Brahmi, both are same medicinal property. But as per a study carried out by CDRI, the Bacopa moneri is more double just medicinal property than Centella asiatica. So in Centella is in place of Centella asiatica, this Merimia emarginata leaves are used in this Centella asiatica leaves. Then symptoms of COVID-19. So now I want to come to this corona, because we have faced a lot of problem during this corona period. So there are 5 symptoms of corona: fever, cough, sore throat, nasal congestion, and running of nose. So in medicine plants, more than 300 medicine plants used for the fever. Acaranthes, Parandrugapis paniculata, Recta indica, Tinospora cordifolia. In cough, 250 plants used in cough, treatment of the cough and cold. So Chorus calamus, Tylophora indica, Justicia datura, Hamidus indicas. Like sore throat, 100 plants, just Piper nigrum, Calimeris, Piper longum, Pipli, Chorus calamus, Mucuna pruriens, Tinospora cordifolia. Then amla is one of the most immunity enhancer fruit. So amla is known to have antioxidant property. It is perhaps one of the richest source of vitamin C. So it is perfect for the overall immunity, and it can rejuvenate and revitalize the body systems. So Chamanpras is a well-known Ayurvedic formulation that can be taken to reduce mental and physical fatigue and boost immunity. So Asugandha is one of the also rasayan. These Chamanpras and Asugandha, Tinospora, these are called rasayan. So Asugandha is also one of the most immunity enhancer plants. Asugandha improves the body's defense against disease by improving the cell mediated immunity. It also possesses a potent antioxidant property that helps the protecting against cellular damage caused by the free radicals. So like Tinospora cordifolia, it is one of the most important medicine plants of our country. Amrita is also known as Amrita in Sanskrit. So a powerful immunity booster due to high amounts of antioxidant, home cure for fever, improved digestion, known to cure hyperacidity, warm infection, a lot of then this is the our Gulchi Tinospora cordifolia. then sajan, it is also one of the most important tree, rather it's miracle tree or mother's breast friend. So it is rich in vitamin A, B, and C, iron, calcium, potassium, amino acids. And moringa also has 46 antioxidants. So more than 300 diseases are cured by this moringa. Leaves are excellent source of vitamins and minerals. So 1 cup of leaves protein, 2 gram vitamin B, say 19%, vitamin C 12%, iron 11%, riboflavin 11%, vitamin 9%. So these are the benefits of the moringa. Did you know that drumstick contains 4 times more vitamin than carrot, 4 times more calcium than milk, 7 times more vitamin C than orange, and 2 times more protein than yogurt, on 0% cholesterol. So these are the medicinal uses of moringa. Then Aishwarya indica is one of itself is a dispensary. A lot of used it is to cure a lot of diseases. Then tulsi, one of the most holiest plant of our country. Every houses has tulsi plants, so it is also very good immunity supporting property it has. And then adrak jejibers officinalis is also one of the most important medicinal plants and plays a very good role in corona. Then haldi, curcuma longifolia, it is also very useful plants. Then kitchen, our kitchen is a medical dispensary. Everybody knows all the ingredients found in our kitchen are medicinal. So thank you very much. Thank you very much for giving me this opportunity. I want to thank the College of Forestry, Banda Agriculture University, and all the organizers involved in this event, this seminar or workshop. So thank you very much. Thank you very much.
Thank you, sir. Thank you for giving us, such a comprehensive and holistic approach about the medicinal plants. And I must say that it was not just about the medicinal plants, the knowledge which you have acquired over decades of your own experience, but you have a thorough knowledge of our Vedas, because you were also, chanting the mantras from the Vedas. So I must say you have an incredible knowledge about the medicinal plants as well as of our ancient history. I'm sure that participant might be having some questions. So dear participants, please, you may ask your questions now. So there is a question, sir. It is believed that to reduce the dependency on the wild stock of the medicinal plants, cultivation has always been recommended. But in practice, it has been, there are substantial evidence that cultivable medicinal plants are not much in practice or in trade. And there is always be a threat, which you also mentioned in your talk, that there is always be a threat to the wild stock of the medicinal plants. So the question is that is there any difference between the chemical composition or the efficacy of the medicinal plants which are extracted from the wild and which are cultivable? So could you please throw some light on that?
एक्चुअल में जो प्लांट्स फॉरेस्ट में ग्रो करते हैं, नेचुरल कंडीशंस में ग्रो करते हैं, उनमें स्ट्रेस कंडीशन में भी वो ग्रो करते हैं. तो जितनी स्ट्रेस कंडीशंस होती हैं उसमें जो मेटाबोलाइट्स होते हैं या जो केमिकल कंस्टिट्यूएंट होते हैं उतने ही ज्यादा बनते हैं उसमें. लेकिन जो मेडिसिन प्लांट्स हम जो कल्टीवेशन में करते हैं वो स्ट्रेस कंडीशन हम उसमें नहीं ला पाते, क्योंकि जहां पानी वो हुआ तो हम पानी दे देंगे, जहां कुछ जो भी कुछ डिफेक्ट दिखाई देगा तो हम उस पर वो करने लगेंगे। तो जो नेचर में जो चीज होती है ना, वो हम कंट्रोल्ड कंडीशंस में उतना ज्यादा नहीं कर सकते। तो जहां तक जैसे मेटाबोलाइट्स की बात है, जो केमिकल कंस्टिट्यूएंट की बात है, तो जो नेचर में जो ग्रो करता है, स्ट्रेस कंडीशन में ग्रो करता है, उसमें मच हायर देन दिस कल्टीवेशन।
Okay. And so what would be your recommendations? The next question is, what would be your recommendations to promote the cultivation rather extracting from the wild?
इसमें दो ही तरीके हैं, मतलब जो कल्टीवेशन है वो तो है ही, लेकिन कल्टीवेशन चूंकि हर प्लांट का कल्टीवेशन नहीं हो सकता, क्योंकि हमारे, जैसे मैंने पहले ही कहा था, हमारे देश की इतनी एग्रो क्लाइमेटिक कंडीशंस अलग अलग हैं कि जैसे हम हिमालय सबसे स्टोर हाउस कहा जाता है मेडिसिन प्लांट्स की, और हिमालय में जो पौधे हैं उनको हम अपने यहाँ पे नहीं ग्रो कर सकते। और जैसे मैंने बताया कि जो अष्टवर्ग की औषधियां हैं, जो 8 औषधियां हैं, ऋष्वक, जीवक, मैदा, महामेदा, काकुली, क्षीर काकुली, रिद्धि, वृद्धि, ये ऑलरेडी क्रिटिकली कंडीशंस हैं, इनके क्रिटिकली कंडीशंस है, मतलब क्रिटिकली इंडेंजर्ड हैं। तो इनका हम कल्टीवेशन कितना कर सकते हैं कि हमारी इंडस्ट्री की भरपाई कर सके। तो इसलिए ये कल्टीवेशन हम उन्हीं औषधि पौधों का कर सकते हैं जो हमारी एग्रो क्लाइमेटिक कंडीशंस में आसानी से ग्रो हो सकती हैं, और उनका जो इजीली उनकी मार्केटिंग भी हो सकती है। क्योंकि आज के बिना जो मेडिसिन प्लांट्स बोर्ड ने, मैं भी उसमें रहा हूँ, करोड़ों रुपए के प्रोजेक्ट दिए इसीलिए कि औषधि पौधों की जो डिपेंडेंसी है वो फॉरेस्ट पे कम करके कल्टीवेशन के माध्यम से करें, लेकिन वो प्रोजेक्ट उतना ज्यादा सफल हो नहीं पाया, क्योंकि उन्होंने बाई बैक पॉलिसी कुछ बनाई नहीं कि मतलब किसान करे तो बेचे कहाँ। तो इसलिए अगर गवर्नमेंट इस मुद्दे पर काम करती है कि वो मतलब पूरा मेडिसिन प्लांट्स कल्टीवेशन के लिए फार्मर्स को जो सपोर्ट करे वो करे, लेकिन उससे ज्यादा उसकी बाईबैक पॉलिसी बनाए कि भाई आप उगाइए, हम खरीदेंगे, और हमारी फिर वो अपना कंपनी को जितने दाम में करे वो करे, लेकिन गवर्नमेंट उनसे खरीदने के लिए रेट तय करके बाध्य हो कि हाँ आप खरीदें। जैसे अनाज की मंडी में हम लोग जाएं तो अनाज हमारा बिक जाता है, भले ही 2 रुपया कम मिले, लेकिन हम खाली हाथ नहीं आते। लेकिन औषधि पौधों में ऐसा नहीं है। जो मैं भी इसमें रहा हूँ, हमने चित्रकूट में नाना जी के साथ में कल्टीवेशन खूब शुरू किया अपने केबीके में, कोई लेने को तैयार नहीं है। अब हम लोगों ने, जैसे मैं एक उदाहरण दे रहा हूँ कि जैसे हम लोग सफेद मूंगफली उगाई थी 2003 में, और कई बीघा में सफेद मूंगफली उगाई, लेकिन बाद में जब बेचने के लिए गए तो जो हमको सब्जी बाग दिखाए गए थे कि 1600 रुपया किलो जाएगी, वो किसी ने 50 रुपए किलो में भी नहीं पूछा। तो ये थोड़े से, ये बहुत से मुद्दे हैं, इस पे जब तक गवर्नमेंट इसमें इन्वॉल्व होकर के कुछ न बनाएगी तब तक कल्टीवेशन उतना ज्यादा सक्सेसफुल नहीं होगा। और फिर दूसरा कारण ये भी है कि जैसे कंपनी को जो माल है, चाहे वो इलीगली ही जंगल से मंगवाती है, तो उसको बहुत कम पैसे में उपलब्ध हो जाता है, तो वो किसान से ज्यादा पैसे दे करके क्यों खरीदेगी। ये भी एक बहुत बड़ा कारण, इसलिए इसके लिए पूरी सी एक बहुत इस पे मतलब पॉलिसी बनाने की जरूरत है, और उसका पूरा इम्प्लीमेंट ढंग से हो तो हो सकता है।
Well said, sir. Well said. So there are some key efficacy issues in implementing the marketing strategy in that plan. Of course, sir.
Definitely.
So yeah, I think somebody's want to ask a question. All right. I would now request Dr. Kaushal Singh, one of the coordinators in organizing this 21-day training program, to kindly deliver a vote of thanks to Dr. RLS Sikarwar. Dr. Kaushal.
Thank you, Dr. Maheshwari. Thank you, sir, for delivering a very comprehensive and excellent lecture and covering so many topics from north to south all over India. And you very easily elaborate all about the medicinal plants, how many medicinal plants used in various traditional medicine systems, and how the various habits of the plants are used which is the highest percentage of plant which is used in Indian traditional medicine system. And you also cover so many systems like Charak, Sahita, and Indian traditional medicine system, endemism of medicinal plants, phytodiversity, medicinal plants for human organs, particularly which medicinal plant is used for the which human organ. And you also elaborate the traditional tribal medicine system. So thank you so much, sir, for giving very elaborate lecture covering the so many aspects of the medicine plants and giving the overview of the whole Indian medicine system or medicine plant used in various medicine system in Ayurveda. Thank you, sir. Thank you.
Thank you very much. Thank you very much for giving me this opportunity. Thank you very much.
Thank you, sir.
All right. So, participants, as for announcement for tomorrow, tomorrow is the first day for the team to forest resources and education. This is relatively a new aspect of forest resources where, two of the very renowned resources talking about certification and the specific utilization of forest. So I'm sure the exact timing would be around 10:30 till 12:30 to lectures would be the schedule would be here and WhatsApp to you by email. So thank you very much for joining us, and I hope you are enjoying and getting trained as well in all the sessions. Have any questions about the program we will ask now and today's session? All right.